so we'll start with Clare Gebbie. Clare's a PhD candidate at Liggins Institute at the University of Auckland. She has a background in genetics and before starting her current studies was a senior technologist at the University of Auckland.
Oh, hi everyone, so I'm Clare. I'm a PhD student at the Liggins Institute and I look at using whole genome sequencing to diagnose rare disorders. So, as we all know, rare disorders, one of the first hurdles for people is often just gaining a diagnosis. And this is particularly true for babies that are in neonatal intensive care. And so often this is a hugely stressful time for patients and families. And in this situation, genetic disorders are overrepresented in neonatal care. So gaining a really fast and accurate diagnosis is hugely important for both families and for clinical outcomes. So the clinical utility of using whole genome rapid sequencing has been shown in over 30 studies since 2012. At the Liggins Institute, we've set up the newborn genomic program to address this gap, and we have a workflow pipeline where patients are recruited through the clinical teams, they're referred to the genetic health service and then recruited into the program. They undergo a consent process and blood samples are taken and sent to us at the Liggins Institute. We do the DNA sequencing and we use a platform called Nanopore which does long read genome sequencing. We then use a couple of different tools for variant creation. And then the results from this are then presented back to the clinical team via a multidisciplinary team meeting that is then fed on to the patients. So we've been doing this for a few years now and we've had 25 acute cases and we've gained a diagnostic rate of 72%. So compared to other whole genome sequencing workflows, this is pretty good. And we've managed to get our turnaround time down to around about 5 days. So this really helps for diagnosing patients. So this QR code here is our first paper that we published that shows our benchmarking results, if anyone's interested. And we've also done some non-acute cases as well, so we've done about 15 of those. So for my research project, one of the benefits of using this long-read sequencing platform is that you're able to gain both the DNA sequence, shown here with the double helix, but you're also able to get another information from that called methylation markers. And this is in addition of a methyl group onto the DNA. And this occurs at sites where there's a C following a G. And this methylation marker is linked to different disorders and human disease. And where methylation marks can occur in certain parts of genes, it's involved with repressing gene expression. So it's also different in different tissues, and it helps if you can think about how the DNA is the same instructions in every part of your body. The methylation can help to control how those genes are being expressed in different parts of your body and at different times. So these, as I say, these methylation marks can be linked to different disorders, and we can test for this using a machine learning classifier. So we can feed the methylation marks from our infants into this classifier, and it can tell us if it can detect 34 different disorders. And so we've done this for all of our cases so far. And this can be useful because it's run independently from the DNA variants themselves. So sometimes you might have a variant where you're not really certain, or there might not be enough scientific evidence to support that that variant is what's causing disease. So sometimes these can be classified as variants of unknown significance or likely pathogenic, or the variant could be, or there could be strong evidence and it could be pathogenic. So we've had 2 cases so far where these were classified as likely pathogenic, so a little bit more evidence is needed to classify them as pathogenic, but they did come back as positive on both the epi signatures. So now I'd like to talk a couple of our cases that we've had. So this case was an infant that came in. They had a few different symptoms, including increased inflammatory response and a rash, and enlarged liver and spleen. And so this infant had, we've discovered a change in the DNA where it should be a C. You can see in the purple box down the bottom. But this one copy of this infant's DNA had a T instead of the C. So this causes a change in the amino acids, which influences how that gene works. So this causes a hyperinflammatory immune emergency. So the syndrome itself is called no-car syndrome, but it can lead to a condition called HLH. I might not say it out loud, but it basically causes too much inflammation in the body by too many activated immune cells. And this harms the tissue and organs in the body. So for this case, this baby was actually able to then undergo a bone marrow transplant to help correct for this condition. So after the bone marrow transplant, the clinicians were interested as to see if the immune cells in the patient's body had been were coming now from the donor, from the transplant, or whether they were still from the infant's original DNA. So they took a sample, what's called a BAL sample, a bronchial alveolar lavage. This is just some fluid that they wash in the lungs, and then so this sample will be a mix of lung cells as well as different immune cells. And we took the sample and we sequenced the DNA. And we could see that about 20% of the DNA was coming from the patient still, and about 80% of that was coming from the donor. But this doesn't tell us the ratio of lung to immune cells. So I then used the methylation marks. We could see that based on the methylation profiles, about 20% of the reds were coming from lung tissue, and the rest were coming from the immune system or other. So this was really useful to show that the bone marrow transplant had been successful, and that most, if not all, of the immune cells in the body had been replaced by the donor. And we did this at 2 different time points. So this is another case. This was our second case, and this was a male infant that had conjugated hypobilirubinemia. In this case, we actually didn't find a genetic variant. But actually, sometimes that's an answer in and of itself. And so this helped the clinicians to determine that this case might be a gestational alloimmune liver disease. So it sort of ruled out some of the genetic disorders that can have the same symptoms. But often just having no result isn't always, you don't know if you've missed something or if that is the real answer. So I looked at the methylation profile of this case as well, and the genes that were differentially methylated between this baby and others were really highly correlated with immune-based mechanisms. So this helped to support that finding that this might be an immune-based condition rather than a genetic variant. So another analysis that I've run for our cohort was using this same method of methylation patterns to look at the different blood cell components in the blood samples that we get. And so the different colours on the plot above here, that shows different types of cells that you find in blood. And then this second plot here shows the differences between all the different cases. And we've identified 3 different outliers. And so 2 of these outliers were the 2 cases that I previously mentioned, which have immune-based causes. And the other one was also Noonan syndrome, which can have a subtype where you also do have myeloproliferation disorder, which can influence the different blood cell components. So thank you. There's a lot of people involved in this project, a lot of different collaborations, Lab Plus, VCGS, the hospital clinical team and genetic counselling services, and I'd also like to thank the funding as well, and of course the families of the patients. Thank you.